the professor focuses on the chromosomal microarray copy number order. So, for delving into the specifics, I am planning to and the first slide is overview. So, chromosomal microarray, or also known as CMA, is a revolutionary advancement in genetic diagnosis that allows for a comprehensive examination of the entire genome at an unprecedented level of detail. So, unlike karyotyping, it is a traditional method which relies only on visual inspection of chromosomes, while CMA uses highly sensitive microarray technology to detect even the smallest deviations. One of its key strengths is the ability to identify minute deletions or duplications of genetic material, also known as CNP or copy number changes. So these CNPs can have profound implications for an individual's health and have been linked to a wide spectrum of genetic conditions. So ranging from diagnostics, offering clinicians, and the researchers' deeper understanding of the genetic landscape, CNPs are there. So by, in, by enabling the detection of CNPs with an parallel position, CMA has paved the way for more accurate diagnosis. So CMA stands at the forefront, driving innovation and ushering in a new era of genetic medicine. So that's how I picture CMA. So for the next slide is the research questions. For the first questions is, can CMA identify underlying imbalances in developmental delay or intellectual disability? And number two, is CMA a more effective diagnostic tool compared to traditional methods like karyotyping, for example, for the conditions like congenital anomalies? So now let's move on, or let's delve into the methodology of CMA. So this CMA harnesses the capabilities of microarray technology, allowing for the comprehensive examination of genetic material. So here are the breakdown of the methodology. So number one is sample preparation. So first is the patient DNA is extracted and fragmented. So according to Miller et al., so the genomic DNA extraction is performed from patient blood cells, while Schaffer et al., the extracted DNA undergoes fragmentation into smaller fragments. Number two is the microarray hybridization. So there are two types of microarray hybridization. We have the CGH and the SNP. So CGH arrays means a patient DNA fragments are labeled with fluorescent dye and co-hybridized with reference DNA on the microarray chip. Differences in fluorescence intensity indicate deletions or mutations for the Schaffer et al., while SNP arrays contains probes for known single nucleotide polymorphisms across the genome. So discrepancies between patient and reference SNP signal deletions or mutations. The next one is data analysis. So here we have the following hybridization. The microarray undergoes scanning to measure fluorescence intensity of each row. Software algorithms analyze the data to pinpoint regions of the genome with gains or losses of DNA. So the last one is the interpretation. So identified copy number variations or CNVs are evaluated based on the criteria such as size, location, and known associations with genetic disorders. So clinical databases aid in assessing the clinical significance of each CNV. Variants of unknown significance or BOUS may necessitate further investigations according to Miller et al. So the next slide will be the critical evaluation. So, so the critical evaluation has its strengths and limitations. So chromosomal microarray has undoubtedly transformed genetic diagnosis. However, a thorough assessment of its strengths and limitations is crucial for optimizing its utility and ensuring precise diagnosis. So the first slide will be its strengths. So here is its strengths. CMA offers superior resolution compared to karyotyping, enabling the detection of minute copy number changes or CNVs across all chromosomes. Analyzing CNVs by chromosome order provides a systematic framework for interpretation, aiding in pinpointing pathogenic abnormalities. This targeted approach enhances diagnostic field by correlating CNVs with known disease-associated genes with specific regions, leading to more accurate diagnosis and interventions. Furthermore, chromosome order analysis streamlines the diagnostic process, focusing on specific chromosome regions with CNVs and avoiding unnecessary testing of unaffected areas. This leads to efficient resource utilization and faster turnaround times for diagnosis. So while CMA has its strengths, it also has its limitations. So interpreting CMA results remains complex, requiring specialized expertise and comprehensive clinical understanding. Distinguishing pathogenic CMVs from benign variants within specific chromosome regions can be challenging, leading to potential misinterpretations and unnecessary patient anxiety. Variants of unknown significance, misinterpretation. Variants of unknown significance, or VOUS, further complicate interpretation, necessitating ongoing research efforts for clarification, according to American College of Medical Genetics and Genomics. Not all CMVs identified by CMA will have a clear phenotypic correlations, particularly for small CMVs or those involving genes with diverse functions. This complexity underscores the need for further investigation to fully understand the impact of CMVs on individual phenotypes. So in conclusion, analyzing CMVs identified by CMA, particularly by considering chromosome orders, offers a potent tool for diagnosing genetic disorder, while it provides a comprehensive view of the genome and facilitates targeted investigations. Interpreting results requires expertise and uncertainties remain with VOUS. Despite these challenges, ongoing research and technological advances hold promise for refining diagnosis and understanding the intricate relationship between CMVs and genetic disorders. So the next one will be its recommendations. So the first recommendations that I have listed is specialized training, because having specialized training means an ongoing training and education for cytogenetic professionals specializing in CMV analysis, which is crucial to ensure accurate interpretation of CMV results and minimize misinterpretations. Standardized training programs in cytogenetics are recommended. Next one is VOUS database. So the development and refinement of comprehensive databases linking specific CMVs to clinical significance are highly recommended. This effort, exemplified by projects like CleanGen, will clarify the role of VOUS and provide clarity for patients and families. And lastly is the standardization of reporting. So standardized reporting guidelines for CMA results, including clear categorization and interpretation of CMVs. It requires consistency, interpretation across clinical settings. So what are its future directions? So I have listed here three things about future direction. So number one is the advanced computational tools. So developing sophisticated computational tools tailored for CNV data analysis can reveal patterns and correlations between CNVs and specific phenotypes. Number two is functional studies. Further research into the functional consequences of CNVs, especially VOUS, which I have stated that it is a complicated topic, is critical for improving diagnostic accuracy and guiding targeted therapies. And lastly is integration with other technologies. So by integrating CMA with advanced genetic technologies like whole exome sequencing can provide a comprehensive genetic profile. Revolutionizing genetic diagnosis and personalized medicine strategies are important. So by implementing these recommendations and exploring promising future directions, CMA analysis can continue evolving as a powerful tool for unraveling genetic disorders, ultimately leading it to improve patient outcomes and personalized healthcare approaches. So that's all. Thank you, Professor Karen Romero, for your attention. I welcome any questions or feedback you may have. Thank you for listening.